Yeah, I guess first and foremost, hi everyone. Thank you so much for being here at 7 p.m. on a Thursday afternoon to learn about Quail, and also to learn about Quail genomics, which for some of you might bring you back to very stressful times in your high school or college careers where you had to learn about genetics. I know it does for me, and I'm working on my dissertation to understand conservation genomics. So that's the first thing. Thank you. And the second thing is I am a fourth-year PhD student at UCLA, and the work that I'm going to be talking about today is a part of my dissertation, is a chapter of my dissertation, and therefore it's work that is ongoing. In fact, I'm trapping and analyzing my data today, currently. And so some of the results are very preliminary, and some of the findings are still require additional analyses and things to tell the full story. So you'll have to, I guess, stay tuned for part 2 of this presentation when I become a finished dissertation student. But until then, thanks for being here. So the outline for today's talk, first, I'm going to spend a little bit of time going into the introduction of what is conservation genomics, why do we study conservation genomics, and some of the background and motivation and questions that we hope to answer with this research study, as well as some of the methodologies that we use to collect the data to answer those questions. Ooh, I'm hearing a little bit of an echo. I don't know if someone has their computer microphone unmuted. Okay, I think it's gone. Second, we're going to go into some of the preliminary findings. Again, I'm going to try to approach this from a very sort of introductory framework, and I'm going to walk you through some of those figures and analyses, but feel free to ask questions at the end if anything was unclear. And then finally, we're going to talk about some next steps for the research to sort of finalize some of the work that we're doing and ways that you all as individuals can help contribute to this research. All right, without further ado, let's get into some introduction. First, before we do that, I just want to take just a brief moment to thank everyone that has been a funder for this project, including San Fernando Valley Audubon, whose work has helped sequence the genomes of some of the animals that we're going to be talking about today. As well as collaborators that have made this work totally possible. There is a small army of people that have been helping to help trap Quail, which is no small effort, as well as to help me analyze these data, which are just crazy and stuff that I never knew how to do before I started my PhD. So very thankful for everyone that's helped, especially so undergraduate students that have helped with this research, and my research advisors and project partners across the Santa Monica Mountains. Okay, so let's get into genome sequencing. So throughout this talk, we're going to be talking about different methods to apply conservation genomics to answering questions about different organisms. And really the sort of beginnings of this work started with humans. And so back in 2001, there was a concerted and collective effort to sequence the first ever human genome. And this effort was multi-institutional, multi-organizational. It was across the entire world, people sequencing parts of the genome and putting these together. It took quite a lot amount of time and also quite a lot of money. So this first human genome, the effort from all these different institutions cost approximately $330 million, which is an incredible amount of money. Cut to modern day, so or sort of today, in 2023, it costs about $600 to sequence a human genome. And you obviously this is several orders of magnitude smaller. And the reason we're able to do this for so cheap is that the technologies that allow us to sequence genetic data have been advancing sort of rapidly over this period of time. On the right-hand side, we see sort of Illumina, which is the forerunner in the development of these technologies. This is their brand new Illumina Nova X or Nova 10 machine that was actually debuted about a month ago. And each time they roll out one of these sequencing machines, they're able to sequence more base pairs of genetic sequence per dollar, and it's just really remarkable. So the sort of cost effectiveness of genomic sequencing paved way in part to conservation genomics that we'll talk about today. In addition, modern genomics is not just human genomes now. The focus is now quite a bit on non-model systems. And when I mean non-model systems, I mean not humans, not domestic animals like cows or chickens, not sort of model laboratory animals like zebra finches, for example. And we can really see that there has been a concerted effort to focus on specific taxonomic groups to make sure that there's equal representation. So some of these consortiums include the Vertebrate Genomes Project, which has the goal of sequencing 10,000 genomes across the tree of life, as well as the Bird 10K project, which is hoping to sequence every bird genome. And other sort of taxonomic focus groups like the BAT1K or the Global Invertebrate Genomics Alliance, all of these have focuses on different taxonomic groups and representation in genomic data, as well as really hyper-local groups like the California Conservation Genomics Project, which actually has funded a chapter of my dissertation that I won't be talking about today, but has the focus of sequencing the genomes of many California ranging species. And so sort of taken in concert, both of these factors make it possible for us to do really interesting and novel conservation science. And really what I'm talking about conservation genomics is using these tools that were developed to study primarily human genomes to answer pressing questions that conservation managers have that would otherwise be almost impossible or very time-consuming and costly to do strictly using observational methods. And some of the questions that we like to answer for conservation genomics include, do these populations show signs of inbreeding? And do we as conservation managers need to take actions as a genetic rescue to help these populations? You can think of like the Isle Royale wolves or the Florida panther, for example. We also ask questions about hybridization. So do we see hybridization between two native species? Do we see hybridization between non-native and native species? And do we need to take action to protect native genes and genomes? We can also use genomic tools to answer questions like, do we see evidence of adaptation across an animal's range to specific climate factors, such as precipitation and temperature? And when we model future climate under climate change scenarios, which are increasingly worse and very likely, do we see evidence for vulnerability in these populations in future climate scenarios? And also we ask questions using genomic tools about population connectivity. So do we see that populations that are separated by different barriers, whether they're geographic barriers or man-made, do we see evidence of gene flow and movement across the landscape? And we ask all sorts of other really interesting and exciting questions. I just mentioned a few here that are things that are on my mind often. And for the sort of purposes of this talk, we're really going to be focusing on the sort of final piece, which is connectivity. So visualized here is a animated GIF of the development of roadway infrastructure here in Los Angeles County. It's a really awesome figure generated by some researchers at Arizona State University. A couple things to point out. This is over time from early 1900s to about 2010. Sort of warmer, redder colors indicate sort of modern construction, sort of greener, lighter colors indicate sort of older construction. And I think it goes without saying, or I think it should be rather obvious that from this image, and I guess also just from looking out most of our windows, that we can tell that Los Angeles is a highly urbanized area. And with these urban areas comes sort of the need to develop roadways and roadway infrastructure in order for us to get from point A to point B, right, to our work, to our families, things like that, which increases our quality of life, allows us to do all sorts of really important things. However, it has the sort of unfortunate consequence of breaking apart the landscape, which can which would otherwise be sort of rather contiguous and which supports a lot of native biodiversity. And so really, we see this sort of interesting sort of interplay of sort of the need for human development and its impacts on native animals. And here in the Santa Monica Mountains of Los Angeles, we have a lot of research on the effects of roadways for our native organisms. Primarily, these have focused on large-bodied vertebrates such as mule deer, mountain lions, bobcats, and coyotes, but we have some evidence of the impacts of roadways on wren-tits, as well as a couple species of lizards. And by and large, what these what these studies show is that there are sort of pretty severe genetic consequences to these roadways, where populations that are separated by roadways show high levels of genetic differentiation and isolation. And there are also sort of behavioral components to these too, where animals will avoid roadways or will shift their activity levels and patterns to avoid human development. Sort of we have a pretty reasonable body of literature to understand what these impacts are. However, we don't know very much about what happens to sort of less agile or sort of less mobile animals, such as the California quail. Quail, I think, are a very interesting system because they defy some of our expectations for birds and how birds relate to roadways. If you can imagine your normal flighted bird, you might be able to just pass right over, right? You might just fly right over the 101 with ease. California quail, however, are a ground bird. You can see that sort of in this GIF right now. But they spend most of their time on the ground to forage for food, to find mates, to lay their eggs, to hide from predators. And in fact, they tend to avoid roadways because they are very sensitive to human disturbance. Some preliminary evidence, such as this quotation on the top, show that in addition to roadways acting as barriers to their movement, when quail attempt to cross these roadways, they can actually act as a high source of mortality too. And so we want to ask the question, what happens to these animals in terms of their genomes? And can we use genomics to understand how they might be moving across the landscape? So if you'll just stay with me for this sort of toy example really quick. If we go back to that sort of map of roadway infrastructure in Los Angeles, we can imagine some sort of theoretical quail populations. So this quail here is sort of bounded in its range by 3 major roadways. And so when it comes up to these roadways, they might act as partial or complete barriers to that quail's movement. So it's not able to find mates on the other side of the road or find new habitat, find food, etc. However, this sort of habitat that is encapsulated in by roadways, it's able to move northward, and there is good habitat north. So this population might be okay, might be buffered against the impacts of this sort of habitat fragmentation. However, when you think of these birds that are bounded sort of on all sides by these roadways and are in areas of very high levels of urbanization, it sort of begets the question, what happens to these birds? And specifically, what happens to these birds' genomes if they're unable to move across the landscape and exchange genetic material? And really understanding what happens to these populations is a major motivating force in my dissertation and some of the questions that we'll answer on during today's talk. So specifically, we have 2 research questions that we're hoping to address with this study and talk about today. The first is there evidence of genetic population structure across the Santa Monica Mountains? And when we talk about this sort of term population structure, what we mean is there evidence in the genetic variation of these animals, of their movement across the landscape and exchange of genetic material, or do these populations look rather isolated? And if they do look isolated, what best explains this genetic differentiation? Is it roadways? Is it some other abiotic factor, et cetera? And our second question is, what are the levels of genetic diversity in quail in the Santa Monica Mountains, and do we see evidence of inbreeding in these populations? And again, we can think of the previous slide where you have that population that is bounded by roadways and is unable to migrate. It's likely that these birds then will end up reproducing with individuals that they're closely related to, and this inbreeding can have really drastic consequences on the health and well-being of these animals. And so we want to see if there's signs using genomic data. And in order to answer these questions, we devised a study to assess the genomes of birds at 6 different sampling locations, visualized here in pink. I should have warned you, there is going to be a lot of pink throughout this presentation, so sorry if that offends your sensibility. But These pink sampling points indicate where we collected quail non-lethally and sampled some of their blood to sequence their genomes. And we have sort of paired sampling locations across the 101 freeway, across the 405 freeway, and across Malibu Canyon Road. In order to collect these samples for genomic sequencing, we first surveyed these areas looking for quail populations. We do this through a visual encounter survey. So that basically just means I go out into the field and I look for quail. In addition, I use a call response survey to locate quail. Sometimes they can be very cryptic and hard to find, which is the story of my life. But they do respond quite well to call response. So you play a call and they'll call back and they sort of let you know where they are. We built custom traps, which you can sort of see visualized in this animated GIF on the left-hand side. In order to entice birds to come into these traps, we baited them with bird seed that you can find at any sort of commercial bird store, including cracked corn and millet. And then once birds were trapped, we removed them from the traps. We took some morphometric measurements, so different lengths, and we assessed them generally for health. And then we took a small sample of blood. And then we fixed a band to their legs so that we didn't sample individuals more than once. And then we sent them back on their way. And from this small sample of blood, we're able to do a variety of analyses to be able to sequence the entire genome of the organism. So the first step really is we take that blood, we bring it to the lab space, which is actually where I'm zooming in from right now. And we essentially do different laboratory protocols to extract the DNA from within red blood cells. We send really high quality DNA that we've extracted from blood to a sequencing facility that runs it through an Illumina machine that we talked about earlier in this talk. That sequencing facility then sends me raw genetic data. So it's just nucleotide bases. And I run those through different analyses and pipelines that my lab has developed in order to be able to assess some of the questions that we're talking about today. Okay. So that was some intro to the motivations and some of the methodologies that we're going to be using. I think now is a good time to sort of focus in on some of the results. So we'll start with the first question. Is there evidence of population genetic structure in the Santa Monica Mountains? And is this best explained by roadways? For this question, we'll be sort of answering it in three parts. And you can kind of think of this as an analogy of, like, for example, a car with a check engine light. You might go to the mechanic, and that mechanic isn't going to just change your oil and say, okay, you're good. The mechanic might do several different sort of diagnostics in order to figure out what's going on with your car. The same is true of genomics, where we apply several different methods. Some of them tell us similar things. Some of them are telling us different things to put together a whole sort of picture. And so we'll talk about three different analyses that we ran to sort of answer this question today, starting with admixture. And so when we talk about admixture, essentially it's a model that you feed genetic variation data and some sort of assumed population. So whether that's just one subpopulation or several subpopulations. And the model assigns ancestry to those individuals. This is a way of sort of detecting whether or not individuals are related to each other. It's a way of assessing do individuals move across different areas and exchange genetic material. I'll just sort of walk you through an example of what an admixture plot might look like and how we might interpret and read those results before we get into some of the real data. So again, there's always going to be a K number, which is essentially just the number of assumed populations. We sampled quail from 6 populations. So today we're going to be talking about assumed populations from 1 to 6. And again, we give this to the model, and the model tells us how well that's supported by our genetic data. We have some proportion of admixture. So that's really just what proportion of the genome of that individual corresponds to one population versus another. Along the x-axis here, we have individuals. So each of these bars represents a single individual and has a unique identifier that I give to these quail. Each of these bars, again, is an individual, and you'll see a mixture of different colors representing how much of their genome corresponds to one population versus another. And then finally, you have the collection location. So here we have W405, which would indicate west of the 405. You'll see different combinations of road side and road sampling area throughout the talk, and that's sort of the nomenclature that you can expect. And again, before we see some data that's real, we're going to think of just some theoretical data of what we might expect to see given certain circumstances. For the first example, we're going to think about a scenario where there is an effect of geographic distance, so how far apart are individuals located from each other, but not an effect of a roadway on the genetic variation that we see across their genomes. For example, say we run this model and it says that based on the genetic variation data, the best supported model is that there's just two populations. And we look at our admixture output and we see really two groupings. We have this green grouping and this orange grouping. And the analysis here tells us that all of the samples on the 405, which are the most geographically distant samples that we collected, all belong to the same general ancestry or relatedness. And those separate out from everything else on the western end of the Santa Monica Mountains, which are colored in orange here, and really just see an effect of how far apart are samples from each other dictates how much genetic variation and difference we see. A different scenario is that there is an effect of both. And I have probably 8 million tabs open, so I'm a little laggy. Okay, there we go. The second scenario is that we see both an effect of geographic distance, so how far apart are samples, and we see an effect of roadway, explaining this genetic variation data. So again, we run our data through this admixture analysis. The model tells us that the best supported model is one where there are 3 populations. And we look at this admixture output and we see that, again, the 405 samples that are really far apart are distinct and are clustering together and are their own color, this green color. And then when we look again at the western Santa Monica Mountains, we see sort of 2 populations, everything north of the 101 and then everything south of the 101, which would indicate that both roadways and sort of geographic distance are important in shaping the genomes and the genetic variation of these animals. Okay. So that's how you would interpret a structured result. And those are some of the sort of a priori hypotheses that we might have when we run this analysis on real genomic data. And then let's see that. So these are the model outputs for our Quail across all of our sampling locations for our genomes that we sequenced for different models from 2 populations all the way to 6 populations. Don't worry if this is like very confusing and there's a lot going on. Sort of the main takeaways here are, one, when we ask this model, what is the best supported model based on our genetic variation data, it says that really through this analysis, there's only one population of quail across the Santa Monica Mountains at the locations that we sampled. And really, based on this analysis, we do not see any patterns of genetic structure across roadways or even through distance in the genomes of these organisms. And there's a couple different interpretations of these results that we'll talk about after we get through the other 2 analyses. But they're, by and large, this set of analysis really gives us sort of one picture. And now we want to assess, do the other ways of assessing population structure agree with this or do they disagree with us? So the next sort of tool in our toolbox is what's called principal component analysis. Principal component analysis is a way to visualize, again, population genetic structure. Before we get into the analysis, I want to take a step back and just think about our data. For example, we send out our blood to a sequencing facility. They send us back genetic variation data. What does that look like? So it comes in what's called variant call format, or VCF format. And what that looks like is this sort of data table visualizes some theoretical data. So it tells us what chromosome a single genetic variant is on. It tells us what position along that chromosome that genetic variant's on. It tells us whether that genetic variant is the same or different than the reference genome that we use to assess genetic variation. And then it gives us this information for all of the samples that we sequenced. And so you can imagine if you have 18 individuals, you have about 17.3 million sort of lines in this data sheet. So it's a really high dimensional data set. It's basically ginormous. And that can actually be really difficult to analyze and to visualize and to try to understand and understand what's going on with the population structure, which is essentially where PCA comes in. PCA is a way of reducing the dimensionality of your data. So basically takes all those different columns and all those different rows and compresses them down into sort of a certain number, a smaller number of data so that it's easier to visualize. For the purposes of this talk, that's probably what we need to take away from that. So it helps us visualize our data by compressing it. And so thinking about what a PCA result might look like, this is just some theoretical data, and I'll sort of walk you through what to expect. So along each of these axes, the X and the Y, we see what are called principal components. These are just one of the, for example, columns of the data that explain a certain amount of the variation in our genetic data. And so we have principal component 1 and principal component 2. And you can see in parentheses how much of the sort of data variation do we explain by this axis. Typically for genomic data, it's quite low, but it is enough to visualize different patterns. And when we plot these two PCs against each other and look at the PCA results, the major takeaway in interpreting this is that points that are close together are more related and share ancestry together. Points that are further apart on either of the two axes indicate samples that are highly differentiated or don't share a lot of ancestry together. Throughout the sort of PCA portion of this talk, you'll see that these points are colored based on both the roadway where we sampled as well as what side of the roadway we sampled them from. Okay, so getting into our actual genomic data. So these are the results of the PC analysis. So I'll walk you through just how to basically interpret this. So along the x-axis here, one of the main takeaways is that samples from the 405 freeway, so those are pictured in the sort of sky blue color here and here. If we look just along the x-axis here, all of these samples are sort of together on the left-hand side, whereas everything else is sort of here on the right-hand side and are all mixed together. And this indicates that this analysis picks up some signal of difference for sampling location along principal component 1, which is explaining most of the genetic variation, which is interesting. If we look along PC2, which is this y-axis here, we also see that this principal component analysis is able to pick apart the signal of side of the roadway for the 405 population again. So we see, right, the east side of the 405 are all clustered here together, and the west side of the 405 are clustered here together. So we're able to pick up that signal and potentially some population structure on the 405 population. However, when we look at everything else, they're sort of garbled together, right? They're all mixed in, and there's no real clear and interpretable pattern of population structure. So just a basic summary is that this analysis is able to see some effect of roadway for the 405 samples, but not elsewhere. Again, we work in concert with all of our different analyses, so we'll do one additional analysis to see what that tells us about the populations in terms of genetic structure. So this final analysis for our first question is called a fixation index, or FST. And you can think of this as just a number that can vary from 0 to 1, which is essentially a measure of genetic differentiation between populations. So you feed it your genomic data, and it gives you this sort of evaluation of how differentiated populations are from one another. You would expect to see that if populations are close to 0, they're not very differentiated from each other, which might indicate that they're freely moving about, they're exchanging genetic material, and there's no real sense of isolation across the genome. As populations approach 1, for this fixation index, or this measure of genetic differentiation, we really see almost complete isolation of these populations, and their genetic material is very differentiated from one another. For wild populations that are isolated and really differentiated, we typically see an FST value of about 0.2 or 0.3 is really differentiated. And populations that are not differentiated are close to 0. Okay, so before we see the actual values, I just wanted to walk through just sort of one toy example again. So if we run this analysis with our genomic data, and isolation by distance is the most important. So basically, samples that are further apart are more genetically distant just because they're further away. You might expect to see that there are really large values of FST between populations that we sample that are really far away when you compare them to populations that are really close together. So you might see higher values of FST between the 101 and the 405 and lower values between the 101 and Malibu Canyon Road. So when we actually run this on real data and see what we get, we see a few things. One is that genetic differentiation across the Santa Monica Mountains is really low. So 0.02 is pretty close to 0. And this value just means that populations might be more freely moving and are readily exchanging genetic material with each other, which is good. We also see that there isn't really any observable difference with this analysis indicating any differentiation between populations. This sort of coincides or corroborates some of the previous findings with the admixture analysis where we don't see a strong signal of population structure. And so when we take all of these three analyses and findings together, we see that generally most of the analyses that we ran support a lack of population structure or a lack of differentiation between quail populations, which might indicate that they're able to move, that they're able to find mates, which were positive things. Although the PC analysis does show us some evidence that there is maybe some genetic differentiation, some fine scale genetic differentiation across the 405 freeway. And overall, there are just generally low levels of differentiation in quail populations. We still have additional analyses to run for this, but that I think gives us a pretty good picture of what we can expect for these birds. So then we're going to move on now to question number 2. So we want to now ask, what are the levels of genetic diversity, and is there evidence of inbreeding in these populations? Before we do that, I'm going to just walk you through what these plots are and how to read them before we talk about genetic diversity. So these are what's called a Manhattan plot because they can resemble skyscrapers, I guess. But essentially, these plots show you along the entire genome by each chromosome, how much genetic diversity do we have at any given location. And you can kind of think of genetic diversity as this measure of how different are the sort of base pairs along the genome. Said in another way, when populations of animals are exposed to different things like disease or environmental disaster, having genetic variation allows potentially populations to adapt readily to an environmental disaster or an outbreak or things like that. As opposed to a lack of genetic variation, you either have, you tend to not be able to respond as readily to these different disasters. And you can imagine sort of if, just like you and I, we inherit half of our genetic material from our father, half from our mother. If our mother and father are actually closely related individuals, it's more likely than not that we inherit at any given location the same, we can say, gene or the same nucleotide, because those people, our mother and father, might have been closely related. And what that looks like when we look at these genome-wide plots is kind of this sawtooth pattern. So we see that there are these long tracks or long runs of areas across the genome where individuals have the same exact base pair or are homozygous for that given location, separated by these peaks in diversity. And so, for example, this is the island fox genome, which is a really inbred island population of gray foxes that are actually dwarfed and were almost to extinction due to a number of factors, including low genetic diversity. When we compare this to a highly diverse population, so this is moose populations on Isle Royale, we can see generally we see an absence of that sort of sawtooth pattern where there's just high levels of genetic variation, which generally means it tends to be good for these populations, like we talked about. Okay, so when we look at the genomes of our quail here in the Santa Monica Mountains, we see a few things. And visualized here, again, are these Manhattan plots, and there's one plot per sampling location. So a total of 6 plots. And we have a couple of takeaways from this figure. One, we see really high levels of diversity, genetic diversity in our quail populations in the locations that we sampled, which again is a really good thing. These are, we'll contextualize it in the next slide compared to other birds, but these are really, really high levels of diversity, which is a good thing. We also see, though, that there is some evidence of this sort of runs of homozygosity here. So we can see one large one right here and one small one right here, which might indicate that in the far past, these individuals did have some interbreeding or inbreeding with individuals that they're closely related with. And further investigations into this inbreeding and homozygosity are things that I'm working on right now, and I hope to share with you in the future. Okay. So contextualizing this now, sort of thinking about heterozygosity, we know kind of why it matters, right? It helps animals respond and adapt to environmental change and disaster. But just taken as a sort of figure, it's hard to really contextualize. So when we compare this level of heterozygosity to some other birds across the bird tree of life, we see some things. So you can compare this against sort of inbred island populations, for example, the great spotted kiwi or the kakapo, which are 2 island populations of birds that are inbred and are suffering some consequences of that. We can see that this value of heterozygosity across the genome is actually quite low. So 0.0008 is a remarkably low value for heterozygosity and genetic diversity. Compared to our quail here in the Santa Monica Mountains, we have an order of magnitude more genetic diversity. This is sort of resembles other sort of populations of quail that have had genomic sequencing and might indicate that these are really genetically healthy populations that are readily moving across the landscape, exchanging genetic material. This is actually a really good sign. So it was very interesting and exciting to see this. So that gives us a sense for the second question, levels of genetic diversity. Now we're going to sort of move into things that I'm currently working on, additional analyses that I hope to run to continue to understand the genomes of these organisms, and finally, sort of ways that you all as individuals listening to this talk tonight can help contribute to this research. So a couple next steps. First of all, we want to sort of investigate. We talked a little bit about these sort of runs of homozygosity across the genomes of these animals. We really want to characterize what extent we see this. So we saw a couple of individuals with these sort of areas in their genome where they lack diversity. We want to ask questions like, what are the genes in those regions? Are they really important genes for the survival of these animals? How common and how abundant are these runs of homozygosity? And what's the sort of timing of this? So are they really long runs that are indicative of really recent inbreeding, or are they sort of shorter runs that are indicative of sort of deep time inbreeding? And these can help sort of answer some conservation management questions for these populations. We also want to take our genomic data and run them through different simulations to estimate historical population size. I haven't mentioned it yet, but generally when people survey quail and have been surveying quail for the past several years, we see that across their range, they're generally declining. So we're seeing each year less and less quail. And so we want to take the genomes that we generated from this study and see if we can detect that decline across their range in the genomes of these animals. And then finally, not related to genetic data, but we have in addition to this genomic data, we've also collected camera trap data from across the Santa Monica Mountains, and we want to use different modeling approaches to evaluate what different environmental factors, for example, drought years, so lack of precipitation, as well as extreme temperature events, as well as urbanization and wildfire across the landscape. How do these factors dictate how abundant birds are at any given location? And we have a little GIF here from one of our camera trap stations that shows male California quail moving across the camera, which is always a good sight to see. Okay, so finally, what can you do as an individual to help this research? So the first thing is, if you see something, say something, which I mean here, if you see quail, let me know. Send me an email. And if you do, please include how many individuals, the time of the observation, and a GPS location. Here's a screenshot from somebody that just randomly messaged me on Instagram and said, I found 8 quail at King Gillette Ranch, and they sent me a GPS location. And this is really a godsend when I get messages like this. So if you're out on a walk, if you're going birding, and you see quail, let me know. For this specific example, just 3 months later, I went to this location, lo and behold, where they found the quail, they were still there, and I was able to trap these birds and collect genetic material. And in fact, this text message helped me collect the birds that were on the east side of Malibu Canyon Road. Second, I'm sure many of you have heard of it, but if you have not, there are 2 community science databases for bird research. So iNaturalist, which you can download as an app or use on your computer, and eBird similarly has a really easy to use app. And essentially what you do is you record observations of animals and give a specific location and time and identify those observations, and these contribute to science. I can say whenever I'm trying to trap quail at any given location, the first thing I do is check iNaturalist and check eBird to see when was the last time someone has observed an animal at this location. And so this is a really great way to help science move forward. And people publish a lot of these results too, which means you can actually help be part of the data collection for a published scientific research project. 3, and maybe this is the most invasive of my asks. If you have a quail in your backyard, let me know and invite me over. I am currently right now trapping quail at the top of Topanga. This is a male quail that I'm desperately trying to get into my traps that comes every morning and sits on this lawn chair. And so if you have occurrences like this where quail are coming to your yard frequently, and if you wouldn't mind, a strange person in your backyard at 6 a.m. in the morning, please send me an email. And then finally, I think a lot of this research wouldn't happen if we didn't have research funding. A lot of this work that takes place at UCLA, we don't have really any support, and we have to solicit things like grants through the San Fernando Valley Audubon. So basically, this is my pitch to say, when you give to organizations like Audubon, you are actually supporting people like me, which are the underfunded graduate student population of the world. And it helps us to do this really groundbreaking and interesting research. So yes. And with that, I will open it up to any questions that you might all have. And then here is my contact information, my email, as well as my personal website in case you want to get in contact with me.
So, Joey, thank you very much. In chat, I ask people to submit their questions by via chat. And we do have a couple that come in from Kathy in particular.
Great.
So maybe, Kathy, you can unmute yourself and ask your 2 questions there.
Yeah, my first one is, I think a question you would ask any researcher: were you surprised by your results? Did you expect this level of heterozygosity?
So to answer your question, yes, I was certainly surprised by our results. We as scientists don't want to influence our results with our expectations, but I can say, as I was designing this research study and reviewing the literature and other organisms, like for example, the wren tit or coyotes, where we did see really strong signals of roadways and genetic differentiation, I was very surprised to see that there is no effect, or at least we did not observe effect with the analyses that we ran. for quail, which are much less mobile and are much more impacted by human disturbance animal. So yes, I was very surprised. And in terms of genetic diversity too, because my expectation was that we would see high levels of differentiation, I also thought that we might potentially see in some of these populations that are entirely surrounded by urbanization, for example, that we might see lower levels of genetic diversity, but overall genetic diversity was super high, which I think was a surprising result, but was also something I was really happy to see, because it indicates maybe that as of right now, at least, there might not be a strong effect of roadways, or potentially we haven't allowed enough time for us to see an effect of roadways, which means that there's time to act. And something I didn't put in this presentation too is that there are conservation actions that are happening right now to ameliorate some of these effects of roadways for other animals. For example, the Liberty Canyon wildlife overpass.
Right. Would you expect to find these same kinds of results in other populations if you sampled in other areas? Or do you think it's somehow unique to the Santa Monica Mountains?
That's a really great question. So there is actually an ongoing research project to assess California quail across the entire state. They only include in their study one sample from the Santa Monica Mountains, but they're really trying to take a broad landscape approach. And so I hope that in the next year, we'll have those results and we'll be able to sort of contextualize what we found in our sort of hyperlocal study with this sort of broader picture. I don't have a strong hypothesis, I guess, for what we would see broadly. In other populations of birds, I can say in the literature, we don't see generally a strong signature of population structure for our like flighted birds, just because they can readily move across the landscape. But it's hard to say for quail. So I'm very interested to see those results when they come out from UC Berkeley.
Okay. Richard, can I ask my second question?
Sure, go ahead.
We live in the Santa Monica Mountains in Sherman Oaks, and when we first moved in the 80s, quail were common. They were all over the place. They nested in our yard. I haven't seen or heard one in over 20 years, but this is an old community, so it's not like there's been a lot of new development. So in terms of population density of people, it's about the same. What we have now are more coyotes. And I wondered, is that why we don't have quail?
That's a very good question. In fact, the way that you sort of told the beginning of that story, like we used to see a lot of quail, now we don't see very many, is actually something I hear from almost every landowner or like homeowner that I talk to, which is a very interesting finding. I think there's a couple things. It is possible that increased predator abundance might have some impact on quail populations. Although basically everything eats quail. That's one of their primary ecosystem functions, is that they're a delicious meal for a lot of different animals, including raptors and other nest predators. So a lot of things eat them. And research actually to like reduce predator populations in the past by TDFW has never really shown to be a big factor in helping populations recover of quail. And I think what the literature points to is more related to climate and habitat. So that's a primary motivation of this camera trap study that we're now developing and working on the analysis for. But largely, it seems that quail are a species of bird that will forego reproduction when there aren't optimal conditions. So quail will just not lay a nest if there, for example, hasn't been a lot of rain and there isn't a lot of vegetation to eat. So that could be one factor that we're seeing hotter and drier years as climate change starts to have its impacts.
I will also point out that we see fewer what I think of as chaparral birds in our canyon than when we first moved in. We used to get thrashers and wren tits, and those are pretty rare. We don't see those at all anymore either.
Yeah, I'm sorry to hear that.
I am too. I mean, we still have plenty of birds. It's just that the composition has changed over the years.
Yeah, and I think that that's probably a pretty shared experience for a lot of folks in the Santa Monica Mountains, unfortunately.
Yeah.
So Joey, I'm going to ask you to unshare your screen so that
Oh,
we
sure.
can see you. I find those quail pictures very attractive, but we're focusing on you right now.
So,
So there's one question from Jolene saying, how many quail would you say you have trapped overall for your research, and how long do you think your research will continue?
ooh, isn't that the $1 million question? I would also like to have an end date for my research and when I'll finish my dissertation. Currently, I anticipate probably having at least one more year of field data collection to catch more quail. To date, we've sampled approximately 120 quail across the Santa Monica Mountains. The study that I talked about today is really just like a pilot study to see some of the patterns that we see in the genomes of these animals. But we also have additional birds from the 101, so not just one sampling location, but several sampling locations on each side. We also have birds from Topanga, and we have birds from Stunt Road. So we're hoping to add those in, as well as sample additional birds in places like the Simi Hills, where there are sort of human development, basically, that are these sort of pocket parks that are totally encapsulated by homes, which quail might not be able to migrate through. So we want to get birds there, as well as other locations like Griffith Park that are really geographically distant too. So we have plans to trap many more quail.
I'm so glad that you want to know about quail sightings, because I live in Woodland Hills and there's a park right by my house that butts up against the foothills, and I see quail there somewhat regularly, a pretty good amount of them. So I'll definitely let you know.
Please hit me up.
So to follow up on that response about looking at places further astray or further away from Los Angeles, I'm wondering about doing something like going way far away, going down in San Diego County, or maybe are there quail on the Channel Islands? Something where you would really expect to have limited interbreeding between them and Los Angeles or San Fernando Valley quail.
I think that that's a wonderful idea. It's something that we're actually working right now with the folks that are doing that statewide sampling project. So I've reached out to them, and we're going to be collaborating. They're going to give us some access to some of the quail that they've sequenced from those areas that you talked about. So different populations in Southern California, but not strictly the Santa Monica Mountains, to contextualize some of our results. So that is ongoing work. There are quail on one of the Channel Islands. So Catalina Island has a population of quail, which are actually thought to be a distinct subspecies of quail. So Callipepla, californica, catalinensis. And those birds are seem to have what's called like island syndrome. So they're bigger, they're brighter, and there's a lot more of them on the Channel Islands than there are in mainland populations. So they, I think, too, would be a very interesting population to counterpoint. And we've actually sampled some museum samples. So we haven't collected birds in the field, but we have museum samples that we're able to extract some DNA from that are potentially going to be sequenced at some point. We just have to find additional funding for that.
So those would be presumably older birds as well. I mean, back in time, as well as perhaps distant.
That's correct. Yeah. So the museum samples tend to be, although there are modern museum collections that are still happening today, museum collections tend to be, older, so 50 years older, which can introduce an effect of time on the genetic diversity you see, which can be something that's hard to disentangle, but can also be really informative to look at adaptation, for example, over time. So yeah, it would add some complexity to the analyses, but might maybe be very interesting.
Okay. So Pat Bates was asking about how you get the genetic samples. You spoke about getting blood samples. I don't know if you get any other tissues, and I presume from the museum birds, you're just getting a skin clipping or I don't know what else, what other sources of genetic material you get.
Yeah, so we're really lucky with the birds that we can non-lethally sample them. So I'm a conservation biologist, so I really want to make sure that I'm not having an impact on the animals that I study. And birds are lucky, or we're lucky that birds have nucleated blood cells. And so there's a lot of DNA in their blood cells, which is different than mammals, for example, which don't have a lot of DNA in their blood cells. And so we're lucky that really just a small sample of blood, so just a couple of drops that we preserve in a buffer, we're able to then take that back to the lab and get tons and tons of DNA. This really helps us with our analyses, and that's how we are able to sequence the whole genomes of these organisms without anything more destructive or harmful to these birds. For the museum samples, you're correct. Bird skins actually maintain a lot of the high quality DNA that we need for this sequencing. So we actually take toe pads from these birds. So we take a little scalpel and we cut off their toe pads, which sounds kind of grotesque, but we're able to then use laboratory techniques to extract DNA even from things that are 100 years old, which again emphasizes the importance of museums and the collections that they do.
So Joanne Milius was expressing her hopeful thinking that maybe you can get extra funding from Sacramento, since California's state bird is the quail.
Yeah, that would be awesome. Maybe fortunately and unfortunately, California quail are what's called a game bird species. And so they're an animal that's readily hunted. And it can be, at the same time, both challenging and sometimes easy to get money for these different game bird species. They tend not to be the priority for funding agencies unless that funding agency has the express goal of supporting game bird populations. So it can be challenging, but sometimes, yeah.
Right.
I have another question too. You know, for the longest time that I've been in conservation, they always talk about crossings over the freeways. Is there anything going on with that yet, or?
Yeah, very exciting news, actually. So the groundbreaking for the Wallace-Annenberg Wildlife Overpass actually took place this year. And they're building I believe, I maybe have this number wrong, $19 billion. Maybe that's too much money. I don't have a concept of money because I'm a graduate student, but they're building a very costly wildlife overpass in Calabasas that will be the biggest in the entire country. And the first time it's been done in Southern California. And so the motivation actually for this research project was in part to assess quail genomics before the construction of that bridge, and then reassess maybe 10, 20, 50 years down the line to see, do we see an improvement in gene flow of these animals? So yeah, that's happening. And also the research might indicate other places where we might need to have wildlife overpasses to ameliorate some of the effects of these things like roads.
Yeah, but for your results, you wouldn't expect to see, I mean, you're not seeing any impact or any significant impact of the roadways on gene flow across in the quail population. Certainly you would expect it in like panther populations, things like that. So in fact, I was looking, you know, the one thing where you had a hint of a result, which was east and west of the 405.
Yeah.
And I'm thinking, wow, there's a huge number of environmental factors which are different across there that might well impact that, independent of migration of the birds. The level of light, the level of insecticides sprayed, the temperature, you know, I could come up with half a dozen more probably of things that could also contribute to a, I don't know, separating or impacting the bird populations rather than just a physical isolation.
Yeah. So that you bring up a really good point. I would say there's a couple caveats to that. One, so this was a pilot study. We only have 3 genomes from any given location. So it's possible that we didn't sample the complete genetic makeup of that population, although we do feel like we have a pretty good sense. Also, we only sampled 3 different localities. There could be areas that we haven't sampled yet that do exhibit some of these signs of genetic isolation. For example, we talked maybe just very briefly about the Simi Hills, right? These populations that are in these pocket parks that are totally surrounded by developments and housing that birds might not be able to get around. So I just want to caveat that with, we maybe haven't sampled the complete picture, and we intend to do more sequencing to make sure that we have. And there are also additional analyses that we'd like to run to just really make sure that the conservation implications of our findings are really clear and are meaningful. So I would tend to agree with you that maybe just based on these preliminary results, we might not see a big impact, but we want to do our due diligence to make sure that we're, really answering the question.
Okay, so we're running pretty long here, but there's one question from Catherine Miller from the California Department of Fish and Wildlife. Is Catherine able to come on, unmute herself and ask that question?
Hi, everybody. This is Catherine Miller with California Fish and Wildlife. Joey and I talked about his project, oh golly, 2018, 2019, I think.
Many years ago.
A little while ago. So I'm really encouraged and really interested in what you've presented. I don't really think I had questions. They were more comments. I'm not surprised at your results, given what I've seen on and what I learned from quail genetic research that I did as well. So that's a good thing. It's encouraging, but to me, it's not terribly surprising. And then I think my other comments were directed at some of the questions about the Channel Islands. I think you answered that really well. We at CDFW in the Upland program have talked about going out onto the Channel Islands and maybe doing some trapping, both contributing to the California Genomic Project, but maybe doing some more specific research projects too. So it's something that's on our radar, but actually going from this is an idea to we're going to do this and we have funding, not just to give other folks, but funding that we can use to do those types of research projects can be a second step that takes a little bit longer.
Yeah.
So, and certainly I know you and I had talked about funding as well. And unfortunately, it just didn't line up that we had an open proposal solicitation notice for our upland game bird stamp. That would be one avenue of funding in the future, but I don't know when we will do our next PSN. So
understood. Well, it sounds like we certainly need to catch up since the last time we talked was 2018, and it sounds like you're doing some exciting work.
Hey, Dr. Miller, since you last talked with us about wild turkeys and stuff, and you also had your colleague present about the Wild Turkey Federation and how helpful they were to helping the turkey population, I'm wondering if there's any sort of game group like that that would be appropriate for helping Joey with his research.
Yeah, so that's a really good question. There used to be a independent organization called Quail Unlimited. And that was struggling financially and ultimately became pulled into Pheasant Forever. So now Pheasant Forever is Pheasant Forever / Quail Forever. And I will talk with the person who is the representative for that about, if he has some ideas or some interest. One of the things that the National Wild Turkey Federation did was to be heavily involved in bringing birds in and moving birds around. And both the representative for the Quail program, the Quail Forever, and I both are, really a little bit more interested in let's figure out how the birds are using the habitat in their native areas and understand that better. So it's a little bit different of an approach than some of the things that the National Wild Turkey Federation does. Quail Forever is heavily involved in trying to promote better quail habitat, absolutely. And I think he would be quite interested in knowing a little bit more about the crossings too, because that's interesting. It poses some interesting questions. I think overall crossings are good. It also provides some questions about if some of the things that are used in the crossings are coyotes and mountain lions, are we driving these animals closer to each other and increasing a potential for predation?
Mhm.
And there isn't really a good way to answer that question. I think that that's a very big, broad, it depends. But it's something that is of interest.
Mhm.
Overall, I think the crossings are a good thing. Not just for the wildlife, but for the cars too. So
Well, thank
So
you.
Joey, I wanted
it's
to
a long
thank,
answer. I'm sorry.
no,
I'll shut up now.
thank you for a very clear presentation. I've seen a number of comments in the chat about how much people appreciated what you had to say. I will tell people that I put your contact information from your last slide in the chat, so you can go there and gather that. If you have a burning question that comes up in the middle of the night tonight, you can send that on to Joey. Or if you feel that the question that you had wasn't adequately assessed. So I just wanted to say thank you again. We really appreciate the ongoing progress and feel grateful that we get to see some of the results that you're getting. Kathy, do you want to finalize things?
Sure. I just want to thank everyone for coming and thank Joey for presenting and for doing the research. It's nice to see some results from our investment. Thank you. And our programming committee has been working hard and coming up with a great series of speakers for next year. So, check back, keep an eye on our website. We've got some great stuff coming up, including in September, Rosemary Moscow, the creator of Bird and Moon Comics and the author of, I'm sorry, what's it called? Pigeons Are Pretty Great. I've forgotten the name of her latest book. And she'll be coming on to talk about her book and pigeons. So have a wonderful summer, everyone. And, keep on birding.